Hello everyone, and welcome to our online lecture series for Bio 1. We're nearing the end now, and in doing so, we've come to probably one of our most interesting and complex organ systems that we'll cover in this class, our immune system, which is a combination of our lymphatic system and the cells that help defend us, generally our immune system. The lymphatic system itself is made of lymph nodes, which do a good job catching all of the water that comes from different areas in our body, mostly from our blood vessels, and filtering it out. So that you can think of this as a filtering liquid system. The organs that you may be familiar with regarding our lymph nodes are things like our tonsils, these kind of ovalish sacs lined with macrophages. We'll talk about macrophages later on in this lecture when we go over the different cell types. And another general organ that you may be familiar with or know as auxiliary is the spleen. It's also a lymph associated with the lymphatic system. It is something that helps, but it's not critical, so you can remove it. Though we're just beginning to uncover some of the potential detrimental sides of removing the spleen itself. Now, the immune system is all about a group of cells that's trying to identify foreign particles or outside particles. Generally speaking, these outside invaders are coming in one of two flavors, either a virus or a bacteria. So let's talk really briefly, just review, what are the two things we're trying to take out? What are the two things that are the enemies that our immune system and lymphatic system are trying to filter from our bodies? Well, viruses are not alive. Though they are very diverse because they need a host to survive. They need to replicate on us, essentially, or any other living thing. They're made of two different components, a protein coat outside and some sort of nucleic acid core. And because of their protein outside, it means that we're going to be using that or specific signatures on the protein coat in order to identify when they are an enemy. Are generally speaking, the things that you can take to help combat viral infections. You can prevent them from ever getting inside of you through reducing spread with things like masks, or a vaccine that gives you an identifier of what the protein coat actually is. We'll talk a little bit more specifically about vaccines when we get down to our discussion of antibodies. You can also combat viral infections with already known antibodies or antibody cocktails. You can, even if your body doesn't have it, you could give it to yourself, even though that's very expensive. And then general antiviral drugs. So either ways to defend once it's already in, or ways that are specific to kill and stop their replication. Bacteria are living cells. They are on their own. They are pathogenic, or can be, meaning that they can make you very sick. They have all the markings of a living cell, so we don't need to discuss their components. They have all 4 of the macromolecules, and they replicate. Because of this, our 2 main defenses for bacteria are antibodies, which in some way are connected to all the different cells that will trigger in order to kill the bacteria. Or, just like antivirals can stop their replication, we can expose bacteria within us to antibiotics, which are meant to stop the replication, stop the cellular machinery, specifically for bacteria. The reason why they're specific to bacteria is they oftentimes target things like bacterial ribosomes, which again, remember, bacterial ribosomes have a size of 70S, not our 80S. So therefore, you can design or find antibiotics that derive from the fungal kingdom to attack these ribosomes, but not ours, which I think is pretty cool. All right, so with an understanding of what we're trying to fight and the organs that we're trying to fight it with, let's go ahead and quickly discuss the structure for what our lecture is going to look like. Because we need to talk about the 2 different components of our immune system. And this will be consistent of immune systems of all vertebrates, so anything with a spine is generally using a similar strategy. We have our innate immune system, and the innate immune system is our first responder. And then we have our adaptive immune system, which is a secondary response. It's a secondary response, meaning that it is much, much slower. But it is very effective, as long as it gets there in time. Our innate immune system is made of either physical barriers or internal defenses, meaning that you can stop the virus or bacteria from getting in on the outside of you, or generally stop it without anything specific, but rather just compounds that are made to try to kill the bacteria once they get in, or virus once they get in. For our physical barriers, you have things like skin and hair and cilia lining your respiratory tract or outside. You have mucous membranes, the salt and water and mucin proteins that line your respiratory tract to prevent things from establishing within cells. You have other chemical secretions in that mucus. You have the digestive enzymes in your mouth that help prevent or break down some things as they enter. And then you have that very effective stomach acid that breaks down most viruses and bacteria that might find their way in there. On your internal defenses, you may be familiar with some of these. You may be not familiar with some of these. You have a general inflammatory response, a complement proteins, phagocytic cells, meaning cells that eat other cells, and your natural killer cells. So let's spend some time now diving into the different components of this, because there is a lot to untangle. We'll first go over the innate immune system and all of its components, and then see how it connects to our adaptive immune system. And I guess I should say before I get into this that it's highly likely that during this lecture you may find yourself needing to pause regularly throughout. There may be a lot of new terminology to you depending on how your background in this topic is. So please make sure that you are utilizing that pause button as we go over the many different components of our immune system. On that note, I don't think we need to go too much into the physical barriers. Obviously, you guys are familiar with the fact that your skin is an excellent preventative measure. It dries things out, plus it's slightly acidic, so it can break down those pathogens. You have other mucous membranes or liquids like tears and mucus that help break down other invaders. Your respiratory system is lined with these things as well, all kinds of different antimicrobial proteins. And stomach acid, obviously, vat of acid kills everything that comes inside of it for the most part. So we'll kind of glance a little bit over the physical barriers, and instead focus on those internal defenses, because those internal defenses are incredibly interesting. We know that these internal defenses of the innate immune system are over 1 billion years old. These things are very, very ancient defenders that are common across so many different organisms. And even nowadays, the ways in which, for example, bacterial immune systems work are so fascinating and so useful to us that it is the bacterial immune system that's the backbone of technologies like CRISPR. We share that ancestry, therefore a lot of this works. We do know that we have our first CRISPR genetically modified humans born on planet Earth, born and alive on planet Earth today. Kind of crazy. The downside to this really old internal defense system is that it is very limited in its number of targets. Meaning that it's not very specific, it's very general. So it's going to do a good job of wiping out kind of things in a certain area, but it won't wipe out everything throughout the body. So you need both of these internal defenses, the innate and the adaptive immunity, and in fact they communicate to one another. When the innate internal defenses are triggered and they identify something that needs killing, they will then trigger the adaptive immune system to follow up. Now, in terms of your innate immune system, there are 3 general cellular responses that you're going to see. They are, number 1, some sort of secreted proteins. Meaning that your cells will identify when something should not be there and try to kill it. Then, there can also be receptor-mediated signaling. Here you can think of things like antibodies. When an antibody binds something that it's identifying as foreign, it then triggers a cascade within the cell in order to bring in all the heavy cavalry and do some digesting of foreign particles. The difference between secreted proteins and receptor-mediated responses is that receptor-mediated stays on our plasma membranes and secreted proteins go out to do the attacking. And then the third response, if you are not able to secrete proteins or mediate a downstream cascade, you then have to communicate to other cells that, hey, guess what? I'm useless, but help. So these are the 3 general cellular responses. And these cellular responses utilize a backbone of cell recognition, because you need to know what you're attacking in order to attack it or make the right thing. That recognition of foreign bodies relies heavily on something called pathogen associated molecular patterns, or PAMP. PAMPs are things like glycoproteins, or glycolipids that are present on the surface of a foreign object. When our body sees these PAMPs, they are identified by a macrophage. Which are our mobile defenders. They move around. And the macrophages are able to recognize things because they have something called a PRR, or pattern recognition receptor. These PRRs are absolutely fascinating. They are incredibly ancient. They have a lot in common with something called toll-like receptors, or they are toll-like receptors. Now, I like the story behind why they're named toll-like receptors. The word in German for great or awesome or good is toll. And because these things were spotted in both developmental biology, meaning that they helped establish our dorsal ventral axis, meaning figuring out what's our top side, what's our front side, and immunity, the scientist working on them exclaimed, "Das ist ja toll," meaning that's amazing. Because these things were involved in so many different components, hence their name of toll-like receptors. So I enjoy that story, and it's kind of interesting to see how many different things toll-like receptors are involved in. They are found both in vertebrates and invertebrates. In fact, their discovery was in fruit flies, so it was in fruit flies that we found that this toll receptor was involved in both figuring out which side the wings would grow on, as well as fungal defense. And they're used in mammalian nervous tissue. So it's actually, the plot is even thicker in that it's not just an immune system thing, but we also use it in nerves. Which, as you get further downstream in studying our different systems, you may see a little bit of a problem between immunity and or using cells similar to our immune system in our nervous cells. It could be something called cross-reactivity. Now, these ancient toll-like receptors are indicative of a very, very ancient arms race, meaning that we've always been evolving the components of our toll-like receptors, and our pathogens will evolve away from it. So it's all about this, I change so I look different, so that way I can identify you. You now will change so that way I can't identify you, but then I'm going to change again, and then you're going to change again. It's kind of like running on a treadmill to stay in place. And this is the way in which toll-like receptors have changed so much throughout time. Now, when a macrophage is going to be recognizing a PAMP from some sort of foreign invader, you're going to get a number of different responses. Some of these tie back to our internal defenses. The first might be an inflammatory response. Now, the inflammatory response is probably something that you're very familiar with. Because anytime that you bump into something, your body might think that, oh crap, something's attacking me, I better inflame a little bit, because you're just trying to increase water and blood flow to the area. And in doing so, you also increase the heat of the region, and the idea behind the heat is that you kill certain bacteria and potentially break down certain viruses. So that way, perhaps you don't even need to launch a total response. You just kill them on the spot with a little bit of blood rush. But just in case, inflammatory responses also recruit something called neutrophils, which are specific cells that will do some killing, so we'll talk about them later. And mast cells that produce something called histamine. And histamine is something that you may be very familiar with because when your mast cells produce histamine, they signal to the rest of the body, y'all might want to get over here, other white blood cells, and start doing some attacking because I think I've got something in here that's probably shouldn't be here. Now, the trouble is that these mast cells are kind of like the boy who cried wolf. They release histamines to potentially too many things that are non-threatening. And you may be familiar with medicines called antihistamines. As allergy medicines. Because oftentimes our allergies are overreactions to things that are not harmful to us, but yet that histamine response causes inflammation and recruitment of other things. Hence the stuffy nose and the secretion of lots and lots of water or mucus. Another possible response, and not one that we're going to go over very much, I'll shrink down the inflammatory section a little bit. Alongside the inflammatory response, you might also have what's called the complement system. And the complement system, again, is part of the innate immune system, but it's just the general idea that there are soluble proteins that the body makes to kill pathogens and liquids. There's a lot more to unpackage there, but not something that we're going to spend any more time on in this class. I just want you to know that it exists. Now, in terms of what are the proteins that are the non-soluble proteins, but rather proteins that are, these are soluble, sorry, I misspoke. What are the proteins made by some of our cells once a PAMP and PRR are binding together? Once this binding occurs, you can get a cascade that triggers the production of proteins called cytokines. Now, cytokines are the first thing made after a PRR PAMPA binding. Oops. Forget whole phrases. So they're the first thing after the PAMP PRR binding, and they help rally the whole body's response. Things that they do that you might be familiar with, they're named because they're part of cell division. So hence the name of cytokines. They are involved in cytokinesis and as well as cell differentiation. And cytokines are responsible for you feeling sick. Your body makes these in order to tell you, yo, we might have something in us, and maybe you should just chill out and go rest under some covers, drink a bunch of water. They make you feel bad or feel sick. Now, these cytokines will induce the production of something called interleukins. And interleukins are named thus because they are the communication proteins for different leukocytes, or white blood cells. And now these are very important and incredibly diverse. There are over 50 different interleukin families that share very, very little amino acid similarity, meaning that these are our method of calling in the right cavalry for the job. They are our signaling proteins that mount the right defense. Now, one of the defenses that they commonly will mount is the production of proteins called interferons. And while interleukins are kind of specific to exactly what they're making, interferons, whoa, let's get a small eraser here. Interferons are a little bit more specific and generally made. What interferons do is they tell all of the neighboring cells to slow down protein production. And think about how genius that is. You don't know which cells are sick, right? You don't know which cells are infected, potentially. But you do know that there are viruses in the midst. So interferons tell all of the neighboring cells, if you know that you're sick or if you don't know that you're sick, start breaking down your RNAs and stop protein production. Because chances are that that protein production is not your own protein production, but instead is the virus's proteins. Alternatively, interferons, if they do identify that the cell is infected, they cause that infected cell to undergo apoptosis. And they also bind to plasma membrane receptors of immune cells, so they recruit immune cells. And in this way, this very long drawn out pathway of a PAMP binding to a PRR on a macrophage will cause the secretion of cytokines. These cytokines will then cause you to feel sick and slow down. Interleukins are a class of cytokines that communicate to white blood cells across the body which ones need to come in and attack. And in the meantime, those interleukins will signal the production of interferons that slow down protein production in the vicinity of the alarm, and then cause sick cells to be apoptotic and recruit other immune players. Pretty crazy, complicated, and cool, but should give you some insight as to how this generalized innate immune system works. The last cell type or major component of the innate immune system that I want to go over are perhaps the coolest named of these, the natural killer cells. And natural killer cells are cells that have a lot of nucleus with very little cytoplasm, because they're doing a lot of processing. They are around to kill tumor and infected cells. So they are a defense against cancer. And this is in the case where the cell itself is able to identify the fact that it is infected. While I would love to go into the ways in which cells can hold up a flag, basically saying, I'm sick, you should probably get rid of me so that way I don't blow up and infect everyone around me, I encourage you to look into it, but it's beyond the scope of what we're going to go into today. These natural killer cells are identifying their targets using something called the major histocompatibility class protein. And there are different MHC groups, like MHC1, which are the ones that they're oftentimes using, and then MHC2s, MHC2s, which are used by helper T cells. More on those in a few. They are really cool because MHC are only present on nucleated cells. So, which cells in your body do not present any MHC class proteins? Well, they're not on red blood cells. So you're not going to mistake or need to screen red blood cells, which is pretty good because your body has a lot of red blood cells and you can just skip screening them. Now, the little bit that I want to explore this idea of cell communication when it's sick and these MHC class proteins, and particularly the MHC class 1 protein, is that your cells that are nucleated will always have these. So they are always present and showing. But if your cell is infected, it can be showing a deformed version. And if the cell is deformed, your natural killer cells will produce two different types of proteins. Perforin, which, as it sounds, perforates and makes holes. So if the cell is sick, you're going to make giant holes in it. And then granzyme, which is going to start to eat the cell that's infected and therefore cause apoptosis. So we just blow everything up, as is the defense mechanism. And now those are the major components and some of the cell signaling cascades that are part of our innate immune system. This innate immune system, if it's identified a foreign molecule in the body that persists or is found in other places, it will recruit the components of the adaptive immune system. So all in all, let's go over all of the different cell types relaying between the two. And I've got red dots next to the ones that I hope you pay particular attention to. As we've talked about already, we have our mast cells and a cartoon image of what they look like. These are the ones that are going to recruit macrophages and neutrophils and cause inflammation by releasing histamines, so involved in allergic responses. We have the macrophages that are the phagocytic, meaning I eat other things, and they are oftentimes some of the first on the scene and will release those different cytokine class proteins. The natural killer cells, which we just went over, they're unique because they move around just like our macrophages. And they're, of course, killing tumors and other virus-infected cells. The cell that is the interlink between our innate and adaptive immune system are our dendritic cells. The dendritic cells are those that present antigens on its surface, thereby triggering adaptive immunity. So these are the bridge from innate immunity, where they've maybe identified something that shouldn't be there, and then they bring it into them, chop it up, and then put all of those broken pieces on its surface. So, in one of my favorite drawings of this entire class, oh, whoops, I gotta change color here. We're gonna go all Mad Max on this. Here you have an invader coming in. The antigen, or the dendritic cell has identified it and has ingested it. Then what it's going to do is it's going to break this sucker apart. Say, you know what? Maybe that's going to be useful. Maybe that's going to be useful. Maybe that's going to be useful. And it's going to display the quote-unquote corpse of this foreign entity as antigens. And in so doing, it will trigger the adaptive immune system to come by and make antibodies. More on that process in a second here. Now, continuing down our list of cells here, we've got monocytes, which are stored in the spleen and move around. They are the precursor to macrophages and dendritic cells. And then we have three different phils: the neutrophils, basophils, and eosinophils. Neutrophils are first responders at the site of infection, and they are about 50% of all of your leukocytes. And they move around to attack anything that is needing to be killed, particularly fungi and bacteria. Basophils are a little bit more rare, and they defend against parasites. They release histamines, just like we see with our mast cells, and may be responsible for inflammation and allergic reactions. Eosinophils release toxins that kill bacteria and parasites, but also cause tissue damage. So you don't want to use eosinophils unless desperate times call for desperate measures. Whew. So you'll have your notes, you'll have this table to refer to when going through your quizzes, because I do know that this is a lot of information, but I at least want you to know where to look it up. Next up, we've got to get into our actual cascade of the adaptive immune system. And the adaptive immune system is known as your learned immunity as well. And no component of learned immunity is more important than an antigen and antibody. So, an antigen is any non-self macromolecule. So it could be a glycoprotein, glycolipid, a specific protein, anything that just identifies you as this particle as non-human. An antibody is the component that's going to bind to it. And when the antibody identifies an antigen, it recruits the immune system to do the killing via eating. Because again, the most effective way to get rid of your enemy is not just to kill him, but to kill him and eat him. Can't waste those calories. At least that's what your immune system is doing. There are two different components to this adaptive immunity. We have our cell-mediated response that utilizes T cells very heavily. And then we have a humoral response. That utilizes B cells and antibodies. Though both of them are using antibodies. These B and T cells have what we call epitope binding. Meaning that they're able to identify the antigen with an antibody. So what is an antibody? Well, this is a schematic of them. They're always made of some sort of transcellular domain, a constant region, and then the very end variable region. They're oftentimes two protein chains, that have a disulfide bridge in between them, giving them a very strong connection. And then at the very end, that variable region is where it's going to bind to the antigen. Sometimes they're shaped like this, as a simple bridge. Sometimes they're shaped more like a Y. But there are other shapes to these different antibody types. Now, I won't be quizzing you on any of these, but it is important to know that they exist and what type of antibodies are possible to work with. So, obviously, we're most familiar with these Y-shaped antibodies, or the IgD or IgE type. And they have these double branching regions that have different variable regions on the end. And of course, the antibody itself is always associated with some sort of signal transduction region, meaning that if I bind here, I cause a downstream effect inside the cell to begin blowing things up. And now, in regards to your B and T cells, your B cells are your antibody makers. And as your antibody makers are therefore part of your humoral response. And I got to correct something I said earlier. I said antibodies are involved in both. That is not the case. Antibodies are present within the humoral response, which is looking at your fluids. So your humors were in Greek medicine, so way back in the day before we knew what we were really doing. We looked at the humors, and by your B cells secreting these antibodies, they are part of the humoral response. Antibodies are not present in the cell-mediated response because instead T cells are doing that work. So it's actually cells identifying things, whereas antibodies are just floating around and identifying stuff to be destroyed. Your B cells are not only the antibody makers, but they're also the antigen presenting cells. And it's in being those antigen presenting cells, or the dendritic cells, because they kind of look like a tree. Forget whether that's a T or a D. Let's check up here. Dendritic, haha, spelling. Dendritic cell becomes your antigen presenting cell in order to communicate with your T cells. Now, your B cells make up about 20% of all of your white blood cells. Your T cells, in contrast to your B cells, again, they work together, but your B cells will call up your T cells. Your T cells are generally considered your attackers and are part of that cell-mediated response. Because rather than free-floating antibodies in your liquids, the T cells are specifically floating to where they need to go and doing the attacking themselves. There are 3 different types of T cells. You have your cytotoxic, which destroy things. Your helper T, that identify infections or other infected cells. And then you have suppressor Ts. They turn off B and T cells. And so let's kind of go over, since we've skirted around this, I want to, in one series, kind of show you all the different steps that happens in an infection. So here we have a virus entering our body, and let's assume that it is not identified by the innate immune system or destroyed by the innate immune system. It will then be brought into your cell via endocytosis. Now, when it's inside of your cell, it's going to be producing its own viral RNA binding to tRNA, to plus strand RNAs and minus strand RNAs. Not super important here, but rather that it's making all of the machinery necessary to copy itself and perform protein synthesis. This will then go through your nucleus and ER and start forming individual protein capsules, and then it will be secreted out of the body and released, out of the cell and released in order to infect other cells. Now, this process will continue up until the cell has literally nothing else to do other than produce these viruses and is so full of them that the cell lyses. Meaning it is so packed and so full of virus that it just explodes in billions of viral particles or millions of viral particles. This infection will continue moving around the body in this way. Now, as the virus invades, you have some chance that it's caught early by the innate immune system, in which case the process of cytokines, interferons, and interleukins would have stopped the infection from spreading. Inflammation would have called in the right amount of backup, or the complement system would have taken this virus out. But in our case here, we wanted to say that that does not happen. So instead, we are going through our cell-mediated response. Because now, this cell here that's a factory for viruses needs to be destroyed. So how does that happen? Well, the virus will float around the body, float around in your humors. It will bump into dendritic cells at some point. And that dendritic cell turns into an antigen-presenting cell and makes that antigen and presents it. So, again, that's the component where it's doing this. Now, when it presents this antigen, it will communicate with a T cell, specifically a helper T cell. The helper T cell will find this infection, release cytokines, and activate the B cells to clone an effective antibody. Ooh, one thing I forgot to mention about your T cells and B cells is the reason why they're named differently is because where they originate. B cells originate and mature in the bone marrow, and T cells originate in the thymus. They mature in the thymus, which is kind of like a barracks. So it's this lymphatic organ right in the middle of your chest, essentially, that holds on to a lot of these T cells because you don't want to release them up until, things have gotten dire. Now, when your helper T cell finds that infection, the B cells will begin to clone antibodies, and the B cells copy themselves clonally. and they begin to ramp up your immune response and communicate to identify the infected cells, so that way these cytotoxic T cells can do the killing. And so you've got this kind of two-pronged attack. Virus coming in, it's identified, the helper T cell is going to arrive on the scene, start some sort of response, hopefully tell the B cells, find an antibody, and attack it while it's floating around, so that way the B cells can focus on the free floating with their humoral response. And then at the same time that the B cells are copying any bound antibodies, because sometimes you need to actually evolve more effective antibodies. And yes, we do evolve and change antibody production in our own lives. So that's the only component of your DNA that's changing. But if they're binding things and hopefully identifying things to kill, then the cytotoxic T cells can go in and identify these infected cells. And so in that way, hopefully that makes sense of your difference between your cell-mediated and your humoral responses. B cells are attacking free-floating viruses with antibodies, and T cells are attacking other infected cells by killing them. Both are triggered by that antigen-presenting cell, so that way you can begin this immune response only when you detect something that needs to be killed. And now that we understand how humoral and cell-mediated responses work, we can talk about antibody concentration throughout time, what this looks like. Once you're first sick, your body might not be making any antibodies. Hopefully your innate immune system's catching it, but in this case, let's assume that it's not. Perhaps in that first Mad Max murder, your body will have some antibodies already on stock that bind kind of okay. That's your primary immune response. Low affinity antibodies start to be made to kill the thing. Let's say that then you get, that will take a while, or you get sick again. Now, after that primary immune response, where the low affinity antibodies are made, your body has learned better how to kill the thing. In a primary immune response, it can take up to 2 weeks to get better. On average, it's taking about 2 weeks. In a secondary immune response, you've already learned what is most effective to kill the disease, and rather than being sick for 2 weeks, you can be sick for something like 2 to 5 days, because you already have high affinity antibodies on tap, ready to kill this free-floating virus that's all around your body. Yes, it got back in, but we know the weapons to kill it. And it is in that learning process, that evolving process, this step here, that you get better at identifying foreign things and you learn. It is this step here that's trying to be fast-tracked with things like vaccines. Because what a vaccine will do is it inserts antigens in some capacity. Either you're making them or they're just exposing you to a large amount of this protein, so that way your antigen-presenting cells can make it. Your antigen-presenting cells present the antigens, recruit the helper Ts. The B cells start working on better antibodies to bind those antigens more effectively. And with things like measles or smallpox or other horrifying diseases, you can actually make antibodies that are so effective that they really do prevent you from getting ill or as ill in the future. And a vaccine is different than the actual disease because rather than carrying that infective lytic process, because as you're blowing your own cells up because of infection, either because they've been identified as infected or the virus is blowing those cells up to spread the virus around further, that's causing tissue damage. And so your primary immune response has more tissue damage. It takes longer to heal from those things. In a secondary immune response, you're limiting the amount of cells that are being infected, responding quicker, responding more effectively, and reducing. You might not even notice that you were really that sick at all. So hopefully that kind of makes sense as to why we have differences in the amount of times we get sick. Sometimes you know what you're fighting with a secondary immune response, and sometimes you need to learn what you're fighting. And again, with things like the common cold, it can take 2 weeks for this. With other diseases like the coronaviruses, it can take even longer depending on the status of your immune system and how similar or how effective your circulation is and all other kinds of things that are largely or partially out of your control. So hopefully with the innate and adaptive immune systems making sense, we'll wrap up the lecture with just a few terms discussing hiccups in our immune system. I've hinted at this already, but allergies are what is called a hyperimmune response. Where your body freaks out to antigens that are not actually threatening. Your body essentially trains to attack something that does not actually cause infection, but because you've identified it as foreign, you will continue to attack it, which is a yikes, and I happen to have a lot of different allergies. Now, this type of hyperimmune response goes through a lot of these same pathways, but you can also have a hyperimmune response to not foreign objects or objects that are not technically foreign. You can have what's called an autoimmune response. And this is the case where you identify self as foreign. And autoimmune diseases are particularly scary, in my most humble opinion. For example, with the autoimmune disease of Guillain-Barré, your immune system may have been triggered to combat the common cold, but in designing its cell-mediated response, it happens to trigger epitope binding that identifies your nerve cells as well. So sure, it's going around killing the virus, but it's also going around and attacking your nerve cells, causing you to be paralyzed, partially, for some time. Now, a lot of Guillain-Barré patients do recover fully, but not all of them. And it, is of no fault of your own, but rather just some sort of response where your body misidentified nerve cells as the thing that's needed to bind to. This can occur because of something called cross-reactivity. Those antigen binding regions might be well suited to bind to a specific region of the antigen, but if that antigen has similarity to receptors on your own cells, now there will be cross-reactivity, which is a yikes. We also can deal with autoimmune diseases with, or encounter ways of combating autoimmune diseases with cases of organ rejection after transplant, where it's still human, but it's not similar enough in your MHC complexes or the 37 other types of histocompatibility complexes, where your body will start to attack an organ that's actually keeping you alive because it's from someone else. In those cases, organ recipients may need to be on immunosuppressors. One, called prednisone, is particularly strong, or prednisolone. I don't actually remember the difference between the two of them, but for example, my dog, when he's having a particularly bad allergic reaction and we can't keep the inflammation down in his paws, will be put on some prednisolone, and he has to pee multiple times in the night. Humans can also be put on prednisolone when they need immunosuppressant activity. And again, those same humans will have to pee many times in the night. I have to say, I will never forget the first time we put Odie on those on prednisolone, and about 2 hours into going to sleep, I heard the most whiny, desperate, "Oh my gosh, I'm about to pee myself, please take me outside," whine from him, and I felt so bad. But I definitely took him outside, and took him outside another 3 hours after that. Poor little dude. Our last topic is on immunodeficiency. Rather than having a hyperactive immune system, there are diseases that can knock out your immune system. The one you're probably most familiar with is human immunodeficiency virus, better known as HIV. And HIV on its own is sufficient to cause acquired immunodeficiency syndrome, or AIDS. And the way this works is that HIV is a virus that will attack your immune cells, your white blood cells, and by attacking the thing that's supposed to be doing your protecting, your immune system becomes very weak. HIV will not directly be the reason why the individual dies, but by having no immune system, it weakens you to a lot of other diseases like the common cold or fungal pathogens. You can get infections where you wouldn't have ever, your body normally fights these things off, but without your your white blood cells floating around and combating everyday infections, those everyday infections now can kill the individual. And so it is very scary if you contract HIV that you may eventually develop AIDS. So HIV being the virus, AIDS being the symptoms that you get from it. But fortunately, through antivirals, antiretrovirals, and other modern medicines, I'm very proud to say that at this point in time, the life expectancy for an individual who has HIV is no different than an individual who does not. If that is not a triumph of modern medicine, don't know quite what is. But again, we've never been able to design a vaccine for HIV up until this point, but we do have therapeutics that can help combat it. So I apologize for the end being slightly scattered here, but hopefully it does make sense as to what autoimmune disorders are, immunodeficiencies are, and allergies are. But again, the most important pieces of this story are the innate and adaptive immune systems that rely on either cell-mediated or humoral responses to knock out diseases in different time frames. It's very important that you understand how the immune system works, so that way hopefully you can do everything possible to keep your immune system fighting effectively.